Today's lecture is going to be talking about evolution, which is how organisms change over time. This is the reason why we live in such a biodiverse world. Organisms are able to adapt and eventually speciate from one another over time. And so we're going to look at the processes that lead to that change. First, though, what we're going to look at is sort of the categorization of organisms. Scientists like to categorize species according to how similar they are to one another. And there's many ways in which this is done. You guys have probably seen something like this in the past where you look at something like a rose, a China rose, which is the species name, but it belongs to a kingdom, right? To Plantae. It has a phylum, a class, an order, a family, a genus, and then finally the species name, which is most specific. So these umbrella categories that get larger and larger and larger as you move up help scientists to identify which species are closely related to one another and which ones are really different. So you can have many different types of species all within one kingdom, and less of those would all be in the same phylum, still fewer would be in the same class, fewer in the same order, and so on until you really get down to just one species or subspecies for an organism. So make sure you know the order of these. Go ahead and memorize those again, the kingdom, phylum, class, order, family, genus, species. One thing I want you to add here at the top is domain. So you'll want to add that in. Because that is really the most broad category, and all organisms fit into one of three domains, and then you drop down to kingdom, phylum, class, order, family, genus, species. Okay, so how do we know who's in which genus and who's in which class? Well, there's a couple of specifications that are used to determine for instance, which phylum something is in. So one of the things historically that's been used is morphology, and morphology is basically what it looks like. So how does it compare to other species in terms of what it looks like? And that was predominantly the way that organisms were classified for these taxonomic purposes. The other category would be behavior. Behavior has been used as a supporting evidence for putting species in certain categories. The new one, though, of course, is DNA technology. So now not only can you see what two species look like and see, well, how different are they, how similar are they? You can think about their behavior, but you can also sequence their genome. The more different the two genomes are in terms of their bases, the A, T, G, C, those orders, the more different they are. So, if I come over here and I give you a sequence for a chicken, okay, so let's say we've got I'm going to use this pen, whoops, see, A, T, G, G, that's for our chicken, and for our dinosaur, which are actually pretty closely related to birds, including chickens, you have A, T, G, C. Okay, and so you would look at the percent difference in bases, and that would tell you how different species are from one another. So there are three criteria that are used to place organisms into these categories: morphology, DNA, and behavior. All right, so that's taxonomy. You can also look at something called a tree of life. So here is the tree of life that is in your book. This is really broad. This literally contains every known organism on Earth in it. And so you start up at the top, and you can see the 6 kingdoms. So here you have bacteria and archaea. Both of those are prokaryotic. And everything else is eukaryotic. So bacteria, archaea, no true nucleus, no membrane-bound organelles. Then you have Protista, Plantae, and then you've got amoeba protistas here, Animalia, and fungi. So 1, 2, 3, 4, 5, 6. It's just you have a divergence here in Protista, so you have it twice. So what do you need to know about this? Basically, I want you to be able to read it. So this is the universal ancestor way down here at the bottom, and this is the cell that all organisms were thought to have evolved from. And then as you move away from that, you get older and older and older. You get newer and newer and newer. So that by the time you get all the way over here to animals and fungi, we are actually the new kids on the block. We are the most recently evolved organisms. So that's one thing, is that you move from old to new. The other thing is finding out recent common ancestors, because that helps determine, well, who's closely related to who. So if you look at this, I know there's a lot of curvy lines here, but if we move to the universal ancestor and we move up one branch point, what you can see is that you still have a bacterial lineage that's coming this direction, but if you move to the right, you have the beginning of eukarya. All the eukaryotes, the protists, the plants, the animal, the fungi. And so this is the most common ancestor of all eukarya. Okay. Another way you could use this would be if I ask something like, well, what is the most recent common ancestor of Plantae? And what you would be able to do is trace that back to Protista. Okay. All right, so that's the tree of life. So what we want to talk about next is how exactly do organisms evolve over time? I'm going to go to a blank sheet for that. Most people associate evolution with Charles Darwin, and rightly so, because natural selection is a really important part of understanding evolution, but there are other ways in which organisms change over time. So let's list those. One would be gene flow. Two would be genetic drift. Three would be natural selection. Four would be sexual selection. And five would be mutation. So I'm going to go over each of these so that you'll have a little background in those, and then we'll do more with this in class. So you can see, though, that natural selection is kind of wedged there in the middle at number 3, and we will get to talk about that, and we will talk about Darwin and Wallace when we do that. Okay, so let's start with gene flow. This is the image that the book gives you for gene flow. It's a nice image that shows essentially two populations of geese. And with this population over here, you have a diverse amount of, well, you have diversity in alleles. You have little a's, recessive alleles, and you have dominant alleles. Over here in this population, that's not the case. So what gene flow is it's any time that you have interbreeding between two different populations. And that's key. It needs to be two different populations. And when that happens from population 1 to population 2, the two populations become more similar to one another. All right, so you're basically spreading the genes out, moving from 1 to 2. And that's gene flow. So that causes a change in this population right here. Okay. Genetic drift. And genetic drift is the second one, and organisms can change over time through genetic drift. This is often associated with negative consequences. If you've heard of bottlenecking, that's when you have something like a mass die-off or a migration of certain individuals within a population, and you have a loss of alleles. So let's use the disaster example. And so what you have up here is flowers, and you can see their genotypes, and there's a diverse array of alleles, right? So look at all of these genotypes: homozygous dominant, homozygous recessive, and heterozygotes. Because of a cold snap, these in the population die off, and these are the only ones allowed to interbreed. And when they breed the next generation, you can see that this is dominated by dominant alleles. That's a problem because you've lost a lot of your recessive allele, and so you've lost a lot of the diversity and phenotype, which can affect the survival of the species. The key here, when you're thinking about evolution, is to understand that genetic diversity is the key to survival, and so that's why genetic drift is often a negative, thought of as a negative event. Okay, so in the instance that everything died off again, and only these were allowed to reproduce, that is definitely a negative situation. You can see here that homozygous dominant, homozygous dominant, you have completely lost your recessive alleles. This is called allele fixation. All you're left with are either all dominant alleles or all recessive alleles. You basically have lost your diversity. And that's very negative for groups, and I'll show you examples of that, some real-life examples of that in class. Small populations are especially vulnerable to this because you can only lose a few and you've completely wiped out all your dominant alleles or all your recessive alleles for a particular trait. So small populations are especially at risk. But that's another way that populations can change over time. Okay, now we get to natural selection, and the grandfathers of natural selection are Charles Darwin and Alfred Wallace. Most people have heard of Charles Darwin, but maybe not Alfred Wallace. They did actually come up with it together, and that's kind of an interesting backstory if you're ever curious about that. There's books out there about that controversy. But essentially what happened here is Darwin discovered on a trip around the world that there were certain postulates for life, and he was able to write about those and publish those, and they really became the foundations of evolution. What you know is that this is one way in which organisms can change over time. In your book, in Chapter 1, it talks about an example of these organisms evolving to become faster. And so this is an example of Darwin's postulates at work, sort of survival of the fittest. You can see a predator here consuming the slow, so that the fast are the ones that are able to breed for the next generation. And so this adaptation leads to more and better fitness over time, and of course is change, and adaptation is change. It also lists all these incredible qualities that this organism has gone ahead and acquired because of natural selection. So camouflage, high running speed, really large eyes, so that they can see 320 degrees. That's pretty amazing. Very large lungs and heart, hollow hair for insulation, strong grinding teeth for breaking up plants, four-chambered stomach for digesting plant food with the help of bacteria, long intestines for digesting and absorbing nutrient-poor plant material. So this is basically saying they can live on very little. They can live on little, and they are extremely adept at escaping predators. So not bad for the pronghorn. All right, so let's break down those postulates before we talk a bit more about evolution via natural selection. So what Darwin's 4 postulates are, if you kind of boil down his theory of natural selection, this is it. And that is, number 1, individuals within a species are variable. That just means that within a population, you have allele diversity that is reflected as phenotypic diversity. And you can see that in skin color here. Eye color, skin color, hair color, all the things we're talking about in genetics. Face shape, all of that. The second postulate is that some of those variations are passed on to their offspring. You know this to be true. You look either a lot like your dad or a lot like your mom. You know that there are certain traits that were given to you from your parents. Those were passed on. The third is that more offspring are produced than can survive, and this really is a critical point here. You've got to start with the variability, but you have to also have some sort of driving force for change. So for humans, things like war, car accidents, premature births. The last is survival and reproduction are not random, and that is that and especially with sexual selection, which we're going to talk about, is that organisms often have a choice in who they mate with, and so they're literally choosing the traits that end up getting passed down, the versions of the traits that get passed down. Okay, let me show you an example of this that's different than the pronghorn. All right, so Darwin's 4 postulates. The first postulate is that individuals within a species are variable. So look at this beetle collection, and these beetles are various colors of gray and black and white, right? So you have genetic diversity. That is the key to natural selection. So the most important postulate here is the fact that you have genetic diversity for natural selection to act on. You have to have the variability in alleles in order to make this work to the population's benefit. Okay, some of that variability is passed on. That would be number 2. Number 3, not all survive. So here you have an opportunistic predator. It consumes the light-colored beetles because it's a black background and it can see them more easily. So what you have over time is a shift to darker beetle color. That's natural selection. One thing about natural selection to remember is that it's very popular to talk about because it always ends in a happy ending. It always leads to more fitness. So if you're a conservation biologist, what you're after is increasing genetic diversity in the population so that natural selection can do its thing, because it always ends in a more fit population because the environment at that time is choosing which traits are ideal and more fit. There are a couple types of natural selection. There's directional selection, stabilizing selection, and disruptive selection. And so examples that the book gives you are, if you had a population of frogs where only large individuals are favored, that would be directional selection. So movement in one direction, only towards the large size, or the inverse would be only towards the small size. Stabilizing selection is where you're selecting for an intermediate size. And you can see the distribution has shifted. This bimodal distribution is disruptive, where you're selecting for both large and small. And I'll show you some real-life examples of this in class. All right, so natural selection can lead to speciation over time. And so you have this image in your book, 17.2. So here you have species A, and you can see that it's been a disruptive selection in terms of these frogs. You have large frogs and small frogs and variously colored frogs. And then if you have this differential reproduction where you're still maintaining these differences and you have some sort of a barrier to reproduction between the two, that can lead to speciation. All right, so this is macroevolution. This is on a macroevolution time scale. And that is, speciation is an example of macroevolution. Microevolution would be like what we are talking about here, these small shifts in the population where organisms can change over time. Once they've become different species for macroevolution, for the most part, to qualify as different species, you have to look at just what we were talking about earlier with taxonomy. Morphology, so they need to look different. DNA, they need to have a certain percentage of their DNA that's different. And behavior. And a big key thing to behavior is that they no longer reproduce with one another. Sexual selection is when you have, usually in the animal kingdom, where you have sexual dimorphism, where the male and female look really different from one another, or the male has specialized structures to call out to females, and females then get the choice of whether or not they want to reproduce. And this can be both adaptive if the females get it right, but it can also be maladaptive if the females get it wrong. So it's risky, but obviously it has persisted in the population. Think organisms like peacocks. And so clearly it has been adaptive because it's still around. The last I'd like to talk to you about as a force of evolution is mutation. Mutation is more of an agent of change. It really doesn't have the ability to significantly affect allele frequencies. When you talk about evolution, evolution basically is change in organisms over time. And what we mean by change is a change in allele frequencies, right? That's what's shifting in the population over time. And so mutation is the agent of change here, right? Mutation leads to more diversity, more allele diversity within the population. And that allows for something like gene flow to act on it, to spread those genes out in the population, if in fact they are adaptive. So mutations can be good, they can be bad. Most of the time they're just neutral and they don't even happen on a gene. Or if they happen on a gene, it doesn't affect the protein. But when they do happen and they are significant, it will end up either being good or bad for the population. So it'll either be amplified in that population through gene flow, or it'll disappear through some sort of a selective force. So mutations really are unique in that manner. They're more of an agent of change than they are a force of evolution. And I'll give you examples of those in class, both good and bad. Okay, so let's go back to speciation for a minute. This is an example of geographic isolation. So we talked about speciation occurs when you have changes in morphology, DNA, and behavior. Here's an example that your book uses on squirrels that were divided geographically, no longer could reproduce with one another, and are now considered separate species. Okay, that's your introduction to evolution. I have one last slide, sorry. This is artificial selection. We're going to do a large unit on this in class. We are going to look at modern day agriculture. This is where humans are the evolutionary force. So what we do is manipulate plants to select for the traits we want. So all of these plants that are in the Brassica family came from this common wild mustard. Another example of this, of course, would be dogs, dog breeds, and that's an example of artificial selection.